Now let us revise the process of megasporogenesis and development of female gametophyte. Single cell inside the nucellus near the micropylar end, it differentiates and it becomes megaspore mother cell. This differentiates and it becomes the megaspore mother cell. The megaspore mother cell, it enlarges in size and it undergoes meiosis. Meiosis results in the formation of 4 haploid megaspores. Out of these 4 haploid megaspore, 3 near the micropylar end, they degenerate and disappear. The last one towards the chalazal end, it enlarges in size and it undergoes 3 mitotic division. 3 times it undergoes mitosis. आफ्टर फर्स्ट ये तीनों जो हैं डीजेनरेटिंग मेगास्पोज हैं. दिस इज द large vacuoles formed in the megaspore, and the nucleus, it divides into two. The two nuclei, they move towards their opposite ends of the cells. After second mitotic division कितने nucleus हो जाएंगे? 2 के 4. After third mitotic division there are 8 nucleus, 4 at each end of the megaspore. Now, this out of the 4 cells at each end, that is 4 at the micropylar end and 4 at chalazal end, इनमें से चारों में से एक एक जो है, one from the chalazal end, one from the micropylar end, they come in the center. They fuse, the two nuclei, the 2 cells, they fuse to form a polar nucleus. The 3 cells at the micropylar end, one is the center, one is the egg cell, बाकी दोनों को क्या कहते हैं? Synergid. And 1 egg cell plus 2 synergids, they form the egg apparatus. And the 3 cells at the chalazal end, they are called antipodal cells, and the cells in the center, which are formed by fusion of 1 cell from the micropylar end and 1 from the chalazal end, they become the polar nuclei, which is diploid, and this results, now the megaspore, it becomes female gametophyte, which is the embryo sac. How you have to write? The out 8 nucleus stage, 4 nuclei थे micropylar end पे, 4 chalazal end पे, 4 में से 3 ने क्या बनाया? एग एपरेटस. एक बना पोलर न्यूक्लियाई, और जो चलाजल एंड पे थे 3 एंटीपोडल सेल्स थे, एक पोलर न्यूक्लियाई. दीज टू दे फ्यूज्ड एंड दे फॉर्म द सेकेंडरी न्यूक्लियस. नाउ during double fertilization, one of the male gamete, it fuses with the secondary nucleus. The process is called triple fusion or vegetative fertilization. It results in the formation of endosperm, which is a nutritive tissue, and the second male nuclei, it fuses with the egg cell, which results in the formation of diploid zygote. This process is called syngamy or generative or the double fertilization, and this zygote, it results in the formation of embryo. Now the next topic is pollination. What is pollination? Transfer of pollen grains from anther of stigma, from anther to the stigma of pistil is called pollination. This was the male reproductive organ, that is stamen. इन्होंने प्रोड्यूस किए pollen grains, and this is the female reproductive organ, that is carpel. Now the transfer of pollen grains from the stamen to the stigma of carpel, this process is called pollination. Pollination is of different types. The first type is autogamy. Autogamy means if the transfer of pollen grains takes place from the stamen to the stigma of the carpel within the same flower, then this process is called autogamy. It is transfer of pollen grains from anther of stigma, from anther to stigma of the same flower. कंप्लीट ऑटोगैमी इज रेयर, क्योंकि नॉर्मल फ्लावर ये सिर्फ वहीं देखने को मिलेगी कि जो फ्लावर्स विच डू नॉट ओपन, जो फ्लावर क्लोज्ड पोजीशन में ही रहते हैं बड की तरह और ओपन ही नहीं होते, वहीं पर ही सिर्फ जो है हम कंप्लीट ऑटोगैमी देख सकते हैं. कंडीशंस फॉर कंप्लीट ऑटोगैमी आर द एंथर एंड स्टिग्मा, दे should lie close to each other so that the self-pollination can occur. For example, it takes place in Viola and Oxalis and Commelina. These produces two types of flowers, self-pollination के लिए या autogamy के लिए. Flowers जो हैं वो दो तरह के हो सकते हैं. एक तो है chasmogamous flowers and another is clistogamous. The chasmogamous flowers, these flowers are similar to the flowers of other species with exposed anthers and stigma. But in clistogamous flowers, the flowers they do not open at all. खुलते ही नहीं है वो. They remain closed. The petals they cover the stamen and the carpel completely all the time. When anther dies, in the flower buds, the pollen grains they come in contact with the stigma. So these are invariably autogamous. तो यहाँ पे सेल्फ पॉलिनेशन के अलावा और कोई चारा ही नहीं है. तो देर आर नो चांसेस ऑफ क्रॉस पॉलिनेशन एंड दे कैन प्रोड्यूस सीड्स even in the absence of pollinators. Clistogamous flowers, what are the characteristic features? They are small, inconspicuous, मतलब वो ज़्यादा क्लियर नहीं है, and they are colorless, they do not secrete nectar. कलर ऑफ द फ्लावर और नेक्टर जो है वो किस लिए होता है? टू अट्रैक्ट द पॉलिनेटर्स. तो इसकी यहाँ पे ज़रूरत ही नहीं है पॉलिनेटर्स की, सिंस दे कैन अंडरगो द पॉलिनेशन ऑन देयर ओन. द सेकेंड टाइप ऑफ ऑटोगैमी इज जिटोनोगैमी. जिटोनोगैमी इज द ट्रांसफर ऑफ pollen grains from the anther to the stigma of another flower but of same plant. यहाँ पे क्या है? The pollen grains they are transferred from the stamen to the stigma from one flower to another but within same plant. देखो, अगर तो pollen grain की ट्रांसफर हो, अगर pollen grain ट्रांसफर होते हैं from stamen to the stigma within the same flower, this is called clistogamy. This is clistogamy. But if the pollen grains they are transferred, if the pollen grains are transferred from the stamen to the stigma of one flower to another flower, means if the pollen grains they are transferred from stamen of one flower to the stigma of another flower but within same plant, then this process is called jitonogamy, and this is also called self-pollination, and it involves a pollinating agent. जिटोनोगैमी it is similar to autogamy क्योंकि pollen grains जो हैं वो same plant के हैं जिनका genetic makeup same है. It takes place in unisexual or monoecious condition where the male and female flowers they are born on the same plant. Now the third type of autogamy is xenogamy, which is also called cross pollination or allogamy. Here in xenogamy, transfer of pollen grains takes place from the stamen of one flower to the stigma of another flower of another plant. It brings the genetically different type of pollen grains and stigma. Now, what are the advantages of self-pollination? The chances of pollination are more. It maintains the purity of race क्योंकि इसमें genetic variation के chances almost nil हैं. Flower need not possess large and colored petals. presence of scent or nectar to attract the pollinators. So self pollination में जो flowers हैं normally they are not brightly colored, they do not secrete any scent or nectar. But what are the disadvantages of self pollination? There are less chances of production of new species, and there are very less chances of variation, and progeny becomes weaker continuously after every generation, क्योंकि वेरिएशन ही नहीं आती है. नई स्पीशीज इवॉल्व होने के लिए जो है वेरिएशंस का होना बहुत ज़रूरी है, बिकॉज वेरिएशंस दे आर द रॉ मटेरियल्स फॉर एवोल्यूशन.